Hi, welcome to the lecture on adaptation for animals. Now we will consider on the hormonal control of circadian rhythm. So beside the suprachiasmatic clock that is giving the neural signal to keep the daily rhythm, we also have hormones that are released only in the darkness. So we have Zeitgeber, light, to keep the suprachiasmatic clock on time. The clock works also in complete darkness and in constant light, but it might run too fast or too slow. And then we have the light that is affecting also on pineal gland that is releasing melatonin. And it's a quite slow signal, as all the hormones are, to compare the neurons. And the consequences are depending that, okay, now you have the hormone all in your body because of the circulation, but then which cells you have the receptor and what kind of signaling cascades there exist in these cells. And to put these clocks a little bit more confusing, it's that they are interacting. So in the lower vertebrates, pineal gland is essential to keep the circadian clock working, to have the rhythm of the circadian clock. But in the humans, it's also affecting. So pineal gland is affecting to put the clock on time, but also on the other direction. So the suprachiasmatic clock is giving signals for pineal gland to keep to release the melatonin at certain time of the day. So where the pineal gland exists? In the human brains, it's difficult to find. It's there behind this hypothalamus, but it's there deep under the cerebral cortex. It's a very tiny organ, just opposite the optic chiasma, and beside the optic chiasma there was this suprachiasmatic nucleus. And in other mammals, it's easy to detect. It's there behind the cerebrum. So it's between the cerebrum and the cerebellum. And you can see that, okay, there is a small two dots, and that's the pineal gland. Very simple to find it. And in both cases, it's located in darkness. It's under or behind the cerebral cortex, under the skull. But in teleost, it's there on the topest. It overhangs over the telecephalon. And for example, in fish and other ectotherms, it's actually, we have two photosensitive organs in the pineal gland. We have pineal organ and parapineal organ. And because they are photosensitive, so they are able to detect light. And that's why the pineal gland in these animals can work even without any eyes. So you can get information, whether it's darkness or daytime or nighttime, directly for this organ. So if it receives the light, we have a very simple photoperiod regulation. And it works even better in the frogs, because in the frogs the pineal organ is connected to the extracranial frontal organ. So there is a frontal organ above the skull under the skin. So it's a third eye. And if you look the structures of these pineal cells in the frog, they look like comb. They look exactly the same as the photoreceptors in the retina. So in these ectothermic vertebrates, several of these pineal gland cells, they contain photosensitive neurons. And they can have different shapes, etc. But so we have photosensitive neurons, okay, then most probably what they are releasing is somehow related on whether it's darkness or light. And for example, in the frog, we have different ways to give this photoreceptor signal. So they can be neurons, So there is a synapse where they are releasing the neurotransmitter. They can be neuroendocrine. So it can be directly getting the signal that, okay, there's light, or getting the neural signal from the other cells, and then releasing small granules containing this hormone, meaning the melatonin, and then it goes into circulation. And this is the way how the human pineal glands is working, although our cells are not photosensitive. Or then it might be the mixture. So the same cell can work also as a neuron and then neuroendocrine. So it releases both neurotransmitter and the hormone. And that makes it very understandable that, okay, the difference between neuroendocrine cell or endocrine cell or typical neuron, it's not so difficult. They are quite same. Okay, how to synthesize and secrete the melatonin. So the pineal gland contains pinealocytes, that are the cells that are releasing the melatonin. It's synthesized from serotonin, that is very typical neurotransmitter in the brains. And then it's released to the circulation, but the melatonin is having a half time for only 10 minutes. So it's metabolized quite fast. So when this pinealocyte stops producing and secreting melatonin, the melatonin concentration in the circulation will drop drastically. And how this metabolized? Well, it's metabolized in different kind of organs. We have in liver, in retina, brains, and pineal gland itself. And so we can have actually quite fast responses with this, because we have so small half time in the circulation. And it's secreted mostly at night time. So the synthesis happens mainly at night, and depending on the light darkness cycle. So if there is only 8 hours of darkness, the synthesis happens for maybe 6 hours, and then if it's 16 hours of darkness, it will take longer time to synthesize. So it's synthesizing longer time, but also more. So it's in higher grade of the melatonin production. But the interesting thing is that, okay, even a short light pulse at the night inhibits this melatonin synthesis. So although you might sleep 8 hours, but if you every once in a while you watch from the your cell phone that, okay, what is the time, you will always drop the melatonin synthesis to 0. And so it's a light sensitive mechanism. That's why it's secreted also is working as in a circadian period. And as a matter of fact, it works so well, so it can be maintained also in the constant darkness. So suprachiasmatic nucleus, where we have this inner clock, it's affecting on this melatonin secretion in completely darkness. So we can have the daily rhythm, but still, if it's constant light, it's not working. So although the suprachiasmatic nucleus now saying that, okay, now it's the time to release melatonin, but melatonin releasing cells are saying no, there's light signals all the time, so it's not releasing. Okay, then if you are releasing a hormone, then you need a receptor. And there are 2 receptors, MT1 and MT2, and they are both working as G protein mediated. They are both G protein mediated receptors, so they are affecting in the cell Y G proteins. And as a monomers, they are both inhibiting adenylate cyclase, so they are inhibiting the production of cyclic AMP, which is a very common secondary transmitter or signaling molecule in the cells. And then interesting is that, okay, these 2 receptors can also work together. And in that case, they are using G proteins, but they are using different G protein to increase the inositol triphosphate and deacylglycerol, so that those other secondary messengers in the signaling pathways. So now, if we have both two different kind of receptor in the same cell, it affects a different process. And beside these, there is one binding site for melatonin in quinine reductase 2, and previously it was called melatonin receptor 3. So it has also, beside these special receptors, it has also some side effects. And actually, because depending of if you have receptor or don't have the receptor, it affects that, okay, whether this melatonin is having any consequences. And so if you have more receptors, then the cells are more sensitive for melatonin, and that's exactly what happens in, for example, in the rat suprachiasmatic nucleus. So in the evening, when it's getting dark, the melatonin is started to release slowly. They are upregulating the melatonin synthesis and release, but these cells are very sensitive, so it gives information quite fast for suprachiasmatic nucleus that, okay, now it's darkness and the melatonin is started to release. But let's see where these melatonin receptors exist in the humans. Over here is a relative expression of melatonin receptor 1 in different tissues in humans, and you can see that it's mostly expressed in salivary gland, testis, kidney, and colon. So most probably melatonin has some consequences in these tissues. Then melatonin receptor 2, it's not in the salivary gland, it's not in the kidney. It's in the placenta, brains, even more in testis, and urinary bladder. So you have different receptors in different locations in your body. And the interesting is that, okay, in the testis there are these both quite highly expressed. So most probably in testis there is also these receptors working together, having different kind of signaling pathways. And then if we check on the side effects that they can have, it's increasing the variability of different organs a lot. And actually all these green are quite much on the related on our intestine organs. So on the stomach or gallbladder or colon or duodenum or etc. So it might have effects on in different kind of organs. And that's why also the variability of the physiological responses related on melatonin are so huge. And so it's lots of binding affinity in several organs. And if we look a little bit closer, usually the melatonin signals and responses in humans are related on the brains. So in the suprachiasmatic nucleus and the different part of the brains, and also in the retina. And then in the lower vertebrates, it's affecting on much drastically on different organs, even outside the brains. It's also affecting on the visual integrating area or auditory system, etc. So it's affecting on the how good we are in seeing and hearing things. And it also affects on these sex-related hormones. So it's inhibiting the luteinizing hormone and follicle stimulating hormone release. So it might be that, okay, that's quite much the signal that in the annual cycle we can have reproduction period at certain time of the day of the year, because at that time of the year we have certain amount of melatonin. There are only two physiological responses that are directly regulated by melatonin. So in the human body or mammalian body, it's affecting on the brains, but they are quite difficult to extrapolate that, okay, what is the direct consequence of melatonin and what is the consequence of suprachiasmatic nucleus or when we are just sleeping. But its effect in the ectoderms on the light adaptation of pigmentation. So it's induced melatonin containing pigment granules, so the frog skin looks different in dark and in the light. And the idea is that, okay, then in the light, the skin color is different, so it's protecting deeper tissues from the radiation. Then the other one is that, okay, it's affecting on the retina, and then we are better to see. It's working in other animals than mammals, and it's affecting on the movement of these photoreceptors. And actually, in these animals, also the retina is synthesizing a very small amount of melatonin itself. And let's look a little bit closer at these. So in the amphibian skill, there are special cells that are regulated in darkness. They have melatonin receptors, and its effects on the movement of these pigment granules. So the skin looks completely different in absence of melatonin and in presence of melatonin. And if there's no melatonin, cyclic AMB is affecting on the depression of these pigment granules. But if we have melatonin, it will inhibit the cyclic AMP, and these dispersed pigment granules will go toward the center of the cell. And when they are all around, the skin looks darker, and that's why it's protecting the tissue from the UV radiation. But it's also inducing movements of the photoreceptors. Photoreceptors are not the first cell to comes in the mind when we are thinking on the cells that are moving, but actually they are. They are a little bit longer in the darkness in animals that are not mammals. And it depends on the melatonin. So it's increased, the length of the cone is increased, and okay, that means that then, because we are modulating these photoreceptor cells in the retina, so perhaps we are getting a little bit better night vision in these animals. But it also might be that melatonin is affecting on the daily rhythms. So in birds and reptiles, these pineal suits are light sensitive, and it's an integral part of the circadian rhythm. But in the mammals, it's not so clear. It seems to be that it affects, if you have removed the melatonin releasing cells, then it causes 4-hour difference in the circadian rhythm. So it's some kind of zeitgeber also for the suprachiasmatic nucleus. So the idea that, okay, the suprachiasmatic nucleus is detecting melatonin, it has to have some receptors. And yes, it has. Over here in the A picture, you can see 2 black dots. It's the suprachiasmatic nucleus, and it's darker because the darkness comes from the melatonin itself. And depending on the concentration of melatonin, then it can't see, etc. So it's a very good indicator that, okay, melatonin has some consequences. It can affect on the suprachiasmatic nucleus. And last but not least, the melatonin is affecting on the seasonal sensitivity. So we get later during this course on these annual cycles, and they are quite much based on the differences in the melatonin concentrations. And that we have two different ways to affect. Either in the long dark nights, the melatonin is released for a longer time, and then we might have larger or different kind of response. Or the other way is that, okay, it can be a phase coincidence. So the secretion is changing night and day, night and day, and night and day. And then if we have more receptors or different kind of receptors, we are getting sensitive. And if they happen at the same time, then we have the response. And actually that's quite much that been tested, found out, for example, in the Syrian hamster. So in the dark period, so in the winter time, they are having a reproduction cycle, after which there's less melatonin receptors, and that's why it's insensitive for melatonin, and that gives 20-week refractory period in this reproduction cycle. So if you don't have the receptors, then melatonin is not having any consequences. So you can modulate your year cycle with being sensitive for melatonin or not being sensitive for melatonin. And for example, here in Eastern Finland, that's pretty much the difference between the winter and the summer. So in the beginning of January, in Joensuu region, we have about 6 hours of daylight. And then in the midsummer, there's maybe 6 hours of not daylight. It's not completely darkness at all. And so we have a large variation, and that makes it a very good signal for different kind of seasonal changes in animals over here. Thank you.